All right. I'd like to welcome everybody today to another of our career advancement series. I am the postdoctoral advancement advisor here at Burke. My name is Edmund Hollis, and today we're going to be speaking with Ali Crawford at Illumina. She has been at Illumina for a decade now. She had 5 years on site before going completely remote. And she runs a lab there and is lead of a translational research team. Now I have full screen, so I actually can't see any of my notes that I had up, but I'll keep that way. So I'm going off memory here. I don't remember you're gonna have to remind me of your title when we get to it, Ali, your current title. But as a graduate student, I overlapped with Ali during her graduate training. I was a postdoc in the laboratory of Yimin Zou at the University of California, San Diego, where she studied Wntz and midbrain axon guidance. And following her defense, she moved to the lab of Joe Gleeson and mapped novel disease genetics and really started getting into genetics and transcriptomics, which is, I think, what she's carried on with at Illumina. So I'm gonna let you go from there. Again, if there are questions, feel free to ask. If you could otherwise keep yourself muted and with your videos off, that would be much appreciated. So Ali, take it away.
Sure. Hi. Thanks, Edmund, for inviting me. I'm happy to be speaking with everyone today. Feel free to interrupt me. I get, like I said, bored listening to myself talk. So just chime in either through the chat or I don't know if it's probably madness if you unmute, but feel free to ask questions as we go along. So I am Ali Crawford. I joke and call it Journey to the Dark Side because I am in industry, but hopefully I can convince everyone that it's not so bad to be a stormtrooper. It's got its pros. It's okay. And so my title, I didn't even put it on here. I'm a senior director of translational research, senior director of scientific research at Illumina, and I lead a translational research team. So I like to start with what I wanted to be as a kid, kind of talk about my journey. All of these things pretty equally, a little bit more or less. So for a while, I thought it'd be cool to be a detective. My parents watched too many crime shows. So that's Stabler and Benson from SVU. Will they get together? I don't know. It's been 20 years. They still are on again, off again. It's a lot of fun. But I thought that was fun. There was a while I really wanted to be a doctor. For you youngins, that is Neil Patrick Harris. He was Dr. Doogie Howser back in the day. So that's a picture of him. I used to want to be an eccentric teacher because I thought that would be fun. So that's Mrs. Frizzled, I think, from Magic School Bus. I'm not going to talk about this, but I also really wanted to be a mail lady. I thought it would be great. I really wanted to drive the Jeep around like on the opposite side of the road. I thought that would be fun, but that dream never came to fruition. And I always wanted to be a scientist. I had a lot of interest in science. I used to mix like different color sodas and pretend like I was doing experiments in our backyard. So I had a lot of different interests, and hopefully I can convince you towards the end of this that I actually ended up with a job, with the exception of the mail lady, where I'm kind of getting to cover a little bit of all of this. So I think that is a lot of fun. I started my research career as an undergrad at the University of Florida. I worked in Dr. Lucia Notterpeck's lab. I'm pretty sure she's at the University of Nevada now, at the McKnight Brain Institute. We focused, pretty much everyone in the lab focused on a protein called PMP22 and its role in Charcot-Marie-Tooth disease. And what was really great about this experience is that Lucia treated the undergrads pretty much like she did the grad students. So I really understood, at least I thought it at the time, what grad school would be like, how to ask a question, how to develop the different experiments to answer that question. So I probably would have told you at the time this was a really challenging experience, and it was, but one I'm actually really grateful for. I learned a lot, and I think it really set me up for my future in science. The other kind of fun thing at the University of Florida was, I was at an institute called the McKnight Brain Institute, which I think was relatively new at that time. I'm aging myself. So I was around a lot of other young PIs that were starting out. We had a lot of postdocs. It was a really nice, vibrant community. After that, I kind of decided, well, what should I do after undergrad? I had an interest in science. I thought had an opportunity to continue it. But I was also a Latin major. That is a thing. I majored in classics. So I was really interested in classical history and in Latin. That hasn't done too much for me for my career yet, except that I can, kind of tell interesting stories. So that that's a pro. And I really like the humanities. So I think it's good to be well balanced. So then from there, I was like, well, myelin seems cool. I applied to grad school, actually at the University of Chicago. I started there early. I worked in Brian Popko's lab. He focused on myelination, and I really thought that's what I would keep doing. I think there's part of me that just felt comfortable with it more than actually really, enjoying necessarily that particular aspect of biology. But when you're in grad school, you do rotations, and my second rotation was with Yimin Zou, as Edmund pointed out earlier. And I ended up joining Yiming's lab. I thought it was very exciting at the time. And then our lab moved from the University of Chicago to UC San Diego. So that was interesting to learn a bit of what it took to set up a lab from scratch. It was a lot of work. I think one thing that was really challenging with that decision was all of my buddies from grad school were obviously remained at the University of Chicago. And so it was very hard to kind of integrate into UCSD because I didn't need to take classes anymore. I wasn't really, like a grad student because I wasn't part of that community. Although everyone was welcoming, I think it's just a bit hard to come in that way. And in Yiming's lab, I had studied exon guidance. Here's some figures I just wanted something to show on a slide from one of the papers that we published from the midbrain. But my focus there was really on like cell culture, cell biology, molecular biology. And so once again, when I finished grad school, which I thought was pretty challenging, I had hard projects. They're technically very challenging. The question of what would come next, I really didn't know. I kind of lost my love for science because of the grind of grad school. It was not a quote unquote easy experience for me. So I wanted to at least give a postdoc a chance. And I really didn't have conviction about what I was going to do next. I just kind of assumed I would still kind of keep floating around in academia, to be honest, but really wanted to give myself an opportunity to have another experience in a different lab with different people to see if I still really enjoyed science. And so I joined during this time, actually at the end of grad school, I got married. My husband loved to surf. I knew we weren't leaving San Diego anytime soon. So I looked for postdocs within San Diego. So it really kind of narrowed my opportunities, which worked out well because I ended up in a great postdoc lab with Joe Gleeson. So Joe was on my thesis committee, so I was well aware of him and had interacted with him before. And this was my first foray into human genetics. And I will say this is where I kind of had my eureka moment. Like, oh man, I love this stuff. I love genetics. All the things that I kind of didn't like in the molecular biology that we were doing, the experiments, the fact that I thought they were, sometimes technically challenging, or, I had to look for like really significant p-values. Like I wasn't necessarily seeing, large biological effects. For what we were doing, at least this type of human genetics, it was very obvious when you had a defect. So in this lab, we focused on a lot of families from the Middle East where you have high rates of consanguinity. So a lot of first cousin marriages. And so it sets you up for kind of the easiest type of genetics to answer questions about disease. So many of these families had children with neurological disorders. Joe would actually go to the Middle East to ascertain samples from them. We had several collaborators in the Middle East, a very gracious community to share these samples with us. And because they had high rates of consanguinity, and we were looking at putative autosomal recessive diseases, meaning that sometimes multiple siblings would have these disorders, it narrowed down our range for what we would have to search for. So we were looking in regions of homozygosity, meaning you got the same copy from mom and the same copy from dad. So it was, like I said, my foray into baby genetics. And we focused on exome sequencing at the time. So I'd never heard of NGS. I had never heard of basically any of the techniques I was doing as a postdoc. So had to learn them. And actually, when I first joined, we didn't even have a bioinformaticist at a time. So I had to learn some of the bioinformatics work. But I enjoyed it. I really, really thoroughly enjoyed it. And, Joe has done amazing things in the human genetics space. He now has probably lots of computational biology students. It was a really exciting time to be in the lab. And while I was in that lab, I'm just showing a figure from, I guess, my major contribution in that lab, which was this paper where we looked at patients with hereditary spastic paraplegia, and we identified novel putative genes from whole exome sequencing doing some network analysis. So we would seed known HSP causing genes in these protein-protein interaction networks and try to predict new ones. And it turned out we actually identified a lot of novel genes in this paper. And the reason that I put this quote here that apparently I don't remember saying, but maybe someone said it for me, but whatever, I liked it. But this is what really did it for me was this part here. We identified several promising targets for development of new treatments. And so this was, like I said, a full circle moment for me. I worked in rare disease as an undergrad looking at Charcot-Marie-Tooth disease. My research has really been around characterizing rare diseases. And now I think there's this foray to like actually interacting with humans instead of just mice or rats and thinking about potential therapeutics. What could be more exciting than all of this? So I was happy in my postdoc, chugging along, wasn't sure what I was going to do next. I had a baby, which was very exciting and new. So I think different considerations with those sort of lifestyle changes. And so I nudged my husband hard, the surfer who likes to surf. I was like, hey, maybe you should get a job that's a bit more stable. He had been a postdoc for a very long time. I think we know getting jobs in academia is difficult, challenging. I wasn't sure that was going to work out for him. And so UCSD had this career and networking fair. This is probably not the one. This is one I pulled today, but they had one 10 years ago, and I wanted him to go. He refused, so I said, "Fine, I'm going to go for you." And so I went, and at this career fair, a person named Stephen Tanner gave a talk about the work he was doing at Illumina. So I was obviously familiar with Illumina because all of our exome sequencing was done with that technology, but that was pretty much the extent of it. And he talked with such enthusiasm about his work that I was really keen to learn more about it. And I went and spoke to him after he gave his talk. And I said, I didn't know Illumina had a CLIA lab. For those of you who don't know what a CLIA lab is, that's a lab that's gone through its validation and verification to offer what's called a lab developed test. And it's doing clinical testing, meaning it's doing tests and it can report it back to a physician. So it can actually go back in a clinical setting and potentially alter care. And as you saw from my previous quote, that kind of aligns with my interest of using genetics to inform precision medicine and therapeutics, et cetera. So when he told me all of this, I was like, this is amazing. I had no idea Illumina did this stuff. This is super cool. And he just happened to say at the time, I think we have an opening. So I emailed him my resume, and I got an email back like, you should apply to this job. And I did. And that's how I got my job at Illumina. It was through networking, quite a bit of luck, being at the right place, I think at the right time, and genuine enthusiasm on my part. When I went into that interview, I had researched about Illumina. I knew what everyone on that interview panel did. I presented my work obviously as a postdoc, which nicely overlapped. It's really one of those moments where I think kind of all the stars aligned, and I started my role there. So then my husband fell off the hook for a little bit, a little bit longer in his job search. But I will say here at Illumina, when I first came on, one thing I want to highlight is that even when you start within a company, just like when you start in a lab, what you end up liking and what you do can change. And so when I first came in, I was working very closely with the software team to develop software to help our clinical lab and others interpret our data. Whole genome sequencing data is complex. You get about 5 million variants from every individual that comes through, and you have to figure out the one or maybe the 2 variants that's causing a genetic disease. Every once in a while, there's more, but for rare disease, it's usually just 1 or 2. So where do you start? How do you do that? How do you make that accessible to someone who is like a genetic counselor or a medical genomicist who does not have a bioinformatics background? So I spent a lot of time working directly with the software, which was one software developer at the time, one person, to develop software that people could use. And very quickly, I realized I do not like that role at all. I like sharing my knowledge, but you become in industry, there have terms like a product owner, where you write requirements, you go through a formal process because you need to test every requirement that comes in. And I didn't really, it wasn't something I was passionate about, that specific aspect. So I actually have done a lot of different things at Illumina. I've had a really unique opportunity there to do that. So I've worked in that space for a bit. I did about a year, I call it internship, but it wasn't an internship clearly, where I worked with the marketing group. I think I made some comments around the fact of how do we prioritize things? This doesn't make any sense. Someone explain this to me. It's like, well, learn some business acumen and understand why certain trade-offs are made or why we prioritize certain things. And that was super, super valuable for me to understand that and to even just kind of speak the language of some of my colleagues. So I was assessing a lot of new technology at the time with the marketing team. And then I've done a lot of different project leads, and I'm still project leads in product development. We built a sample to answer solution in the transplant diagnostic space. So these are folks getting either an organ transplant or a bone marrow transplant. And so if you hear you need to be a match to donate, referring to HLA typing. So we developed an NGS assay for HLA typing. I knew nothing about HLA, and my goodness, if you want to look at, if you want to feel stupid, study immunology. Holy cow! Like it's constantly changing, super complex. HLA region in the genome for genetics is extremely polymorphic. It's extremely hard to resolve. So we had to develop kind of novel assay library prep techniques to be able to do that. And you needed to provide the software to be able to understand and interpret those results. So it was a great experience because it was really soup to nuts, like sample in, report out. We worked with KOLs in that field. Very exciting time. And then the other project that we worked on related to that was in chimerism. So chimerism testing is what I would call like a liquid biopsy test. So in the case of a bone marrow transplant, when you get someone else's bone marrow and then someone does a blood draw and they look at your DNA, you now have the DNA of the donor, not yourself. So if you're ablating all of your bone marrow, don't use that for evil. But what you can do if you want to know if that graft is failing, if you see the recurrence of your DNA coming back, that means that bone marrow transplant is not doing well. So it's something called chimerism testing, or you've probably heard of things like minimal residual disease. So we built a really cool liquid biopsy assay to that. And the other way that you can use that, it's kind of the flip side. If you get an organ transplant from someone, and if you look at the cfDNA, so cell-free circulating DNA, and you see that person's DNA popping up in your blood, that means your organ is failing. So this is so impactful in this space because the way that we do transplants today is that someone needs a kidney transplant. You put this person through hell, right? You give them immunosuppressants, they go through this surgery, you take this beautiful pristine kidney, and then to know if it's doing well, then you force this poor individual to go in and get it poked and do physical biopsies to check the health of this organ. It's kind of crazy, right? So to have non-invasive ways to do that by looking at cell-free DNA really, I think, transforms the field and makes a much better patient experience. So long story short, my background was in germline genetic disease. I came in, I got to learn marketing. I got to learn about our immunogenetics assays, HLA typing, transplant diagnostics, liquid biopsy. And then since then, I've done two different things. So I lead a product development effort on whole genome sequencing in the clinic. So trying as much as possible to be compliant with different regulatory requirements in this space and to make it as easy as possible for customers to adopt it. So fully automating it, doing all the bioinformatics behind it, et cetera. And then my other job, which I'm gonna take the remainder of the time talking about, is I lead our translational research group, which is my favorite and I love it. So I work within a group in the company called Medical Genomics Research. Our focus, if you were to sum it up in one line, is to demonstrate what is possible. So our remit is to try to understand where our technology and potentially other technologies could be used in the clinical space and try to be really kind of forward thinking. And so the idea then is that we can take those lessons learned, bring them back to the company and suggest, hey, we should invest here, we can improve our products here, et cetera. And so we do that through kind of 3 different ways. We solve rare disease cases that come through. Sometimes they come through our clinical lab, but we don't get an answer. They consent from research, and then we do other things for that case. And through other collaborators, other PIs that reach out to us that have collaboration interest. So we get to solve cases. So patients that previously have some unresolved, putatively we assume a genetic disease. We are the opposite of a lot of people in the industry. We want to publish everything that we do. We want to get it out there because we want to move the field forward. So we want to make open source tools. We want to put all of our findings out there. And then the other fun thing we get to do is prototype. So we evaluate new types of analyses, new assays, and sometimes different technologies in this space to inform the group. Lots of fun. And if you remember all the things I told you I wanted to be when I was a kid growing up, it kind of like hit the check boxes here. So you get to be a gene detective, and that you look for novel genes to solve cases. You're, informing a potentially future therapeutics for this patient by providing them a molecular diagnosis. We get to publish and educate the community about what we do. And then, we get to do fun things in the lab and prototype and to be a scientist. So I somehow managed to find a job where I could do everything but be a mail lady. That's just one dream we'll have to cross later in the future. So just to give you a little bit of background about kind of what we do in the rare disease space, for those that don't know, there's probably about 250 million people in the world that have what we call a genetic disease. Very few people have access to genetic testing. Very, very few. I think it's, well under 1% across the world. And then those that do, they often only have a diagnostic rate of about 50%. And so our big question is, what are the additional tools and methods we can do to try to diagnose the remaining 50? And so we have ongoing now a what we call an observational prospective clinical trial. Observational means we're not giving drugs, we're not in any way impacting the treatment, but we are observing what these the additional tests that we do, how that can provide answers for these families. And so that study is called I Know. You can look it up. It's actually in clinicaltrials.gov. And this is why we have this catchy slide called I Know Omics, where we have whole genome negative families. So they've gone through, got a whole genome, didn't get a definitive diagnosis. And we do all the assays we can think of to try to find answers for these families. It's super, super rewarding. It is a lot of fun. And I know I'm very privileged to be in the position to be able to do this and have access to these sort of technologies. The other great thing about this particular study is, as we said, as we went into it, we absolutely want to make all this data available as a community resource. And so we are working with the Gregor Consortium and other ways to make that data available so researchers can look and have kind of a benchmark set or find things that we don't find. So we've been successful in finding some of the answers, but we haven't solved all the cases. And our goal is always to try to find an answer for every single participant that comes through. It's a lot of fun. So if you want to talk about the science stuff, let me know. I can do that forever. But now I want to talk about you. Go ahead.
Can I interrupt with one quick question,
Yeah.
Ali? So just out of curiosity, I think this I Know Omics idea is like awesome. You're basically doing every possible thing to figure out. But how many would you say, rough estimate, of the 50% that didn't get anything, once they enter this incredible pipeline, do you get another 50% answers, or what's your success rate?
It varies a lot on the phenotype. And so you have to think about things like tissue accessibility. So for example, if you have an immune disorder, using blood as a sample is actually pretty good because the disease is in the blood. And so for things like those sort of disorders, whole transcriptome might prove to provide some insight. Whereas if you had a gene that's only expressed in muscle, you would need to do a muscle biopsy to see changes with transcriptomics. So I would love to give like a very definitive answer, but I think it really depends on the phenotype. I've been surprised with things like methylation has actually been really useful for us. We work with a group called Episign. They have a great test, and we've been able to diagnose some of the neuro-related phenotypes with methylation. And we're still learning. I don't think we have developed the tools or the knowledge yet to fully utilize all the information. It is a lot of, this is like a data set you can analyze for years that's coming too. But even a 10% bump is very significant. I think a huge portion of what's going on is just, you can do all the tests in the world, but if you don't understand the biology, you're going to miss a signal. So, in genetics, you have things like partial penetrance. You don't always know the environmental factors. I think those other things will come into play. There's a lot of AI tools that are being developed in this space. Alpha Missense just came out. Illumina has this primate AI 3D. All those things will help, I think, in our interpretation of the biology. It's one thing to get analytes, but if you don't know what they mean or how to like link them together, I think that's kind of the next big step. Oh, thanks for the question. Okay, like I said, enough about me. Now about hopefully everyone on this call, to really kind of think about a couple of things. This would be kind of my recommendation to you and how to be thoughtful and purposeful about what you do next. And I think it's important to take the time to think about what it is that you really want to do. What are your values? What kind of work-life balance you may need? There's people that are caretakers that might have elderly parents that have to live in certain places. There's all sorts of different considerations for individuals. And really think about, why did you do a postdoc? And do you think the skills and the connections that you've made will ultimately take you where you want to go? I think we often end up on a path and fear deviating from that path. And my message to everyone is like, most routes are circuitous, and I think it's important to be open to that. And the fact that you have these sort of seminars, I think, shows that there's a lot of support for the postdocs in your community, which I think is fantastic. You should use that support. You should talk to each other. And this definitely doesn't mean don't go into academia. That is a lot of people's dreams, and I think it's fantastic. And I have friends in academia that absolutely love it. But there's a lot of different things that you can do. And I think we all know biotech, everything is hard, right? Getting a job these days are hard. So I think it's important to kind of open it up and ask these big questions. Don't think about what I want my next job to be. Think about it, what do I want to do? It's two different questions. It's don't think about title. What is it I want to do? And the other thing that I did that I thought was really useful for me, that I recommend to everyone else, is these sort of Venn diagrams. If you can find this sweet spot where you're working on something you're passionate about or you enjoy, you're good at it, and it can make you money, you've hit the jackpot. Like that's ultimately where you want to be. And once again, this isn't title based. This is a thing based, a doing based thing. The other thing, and I don't know if this has ever been presented to you, is something called individual development plans. I love the date on here, 2014. It's been a while, but I actually did one of these and went through it and did everything on this plan, and it really helped me a lot. I didn't necessarily appreciate it as a useful activity at the time, but if you haven't done one, it really makes you think of kind of like your career goals. Like I said, it's kind of focused on position, but I think it should be focused on what you're doing, your contribution, and kind of your action plan to get there. And I think discussing that with your PIs, or if you have other mentors to your network, I think it's really, really useful activity. I cannot emphasize it enough. So if some of you are interested in joining the dark side, which I joke is industry, hopefully you see that we actually do good things. We actually benefit patients, and we're moving it forward. But I will accept that many of us refer to us as the dark side. I just want to take the last few minutes to kind of talk about a few common myths and what I think you should consider as you're moving in that direction. I've been shocked at the number of people who apply to be a scientist who don't want to do bench work. I just warned you, if you're applying to a scientific position, you're absolutely going to be at the bench. It does not matter. You should make it very clear up front what your expectations are. I've been really, really surprised by that. Getting a job in industry is easy. Okay, it was for me. I got really lucky, to be honest, but that was not the norm, and it's extremely competitive. Lots of very, 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 very talented people are applying. That being said, a lot of people don't apply because they don't think they have the right qualifications. And I guarantee you, if you've been a postdoc, you have done so many skills, and I'll talk about translational skills in a moment, that makes you amenable to industry positions or any position, even outside out of industry. We just don't always know how to kind of sell ourselves in that way or present our skills in that way. And then I hear a lot, of course, I think I've probably convinced you otherwise with my background. Industry jobs are boring and kind of you lack, creativity. It's not. I really am blessed to work at a very, very innovative, exciting company with super highly motivated individuals, and it's lots of opportunities for creativity. So none of these things are true. There's probably other myths and misconceptions I'm happy to walk through, but these came up quite a bit. But really do want to emphasize that the right qualification. You don't have to have every single qualification to apply to a job. Very few people do. So don't hold yourself back there. So what can you do with a PhD in industry? I just want to highlight, and I just put something on here, a lot of different things that I think maybe people aren't aware of. So obviously the actual product development aspect and research is obvious, but we have great technical writers, graphic designers that go into industry, if that's an outlet for you, using your scientific background. And if you have artistic abilities, that will get you far. That is always something that's needed. All sorts of technology transfer. Obviously, if you're a very outgoing person, sales, and everyone's like, oh, sales is bad. No, sales is not bad. Like getting our technology out there that I think is life-saving is really important. And if you don't, people have to know about it to be able to purchase it. So I think our sales teams are amazing. They do fantastic work. Project management, doing a PhD is tons of project management, right? Lining up what experiments you need to do, sometimes delegating those efforts, coming up with timelines, all of those things like are managing large projects, especially if you've written any sort of grants and had funding as well. That's even kind of a bigger program purview. So definitely do not, people sell themselves short. Don't sell yourself short there. A lot of people go into things like clinical trial management. Once again, this is something you can segue into. I know people that have gone and actually got their went back to law school and got patent law kind of positions, which is great because once again, you need that kind of scientific background sometimes to be able to do that. I wouldn't. It's very hard. I don't understand the law at all, but for other people, it works for them. Lots of quality roles, regulatory affairs. All of these things are open and possible for folks with scientific backgrounds. And then the last one I will mention, scientific policy. There's folks that actually lobby on the Hill for different access to different tests, for example, which I think is super exciting. Consulting comes up all the time. Scientific and medical liaisons and different sort of analysts. So just want to highlight there's all sorts of things that you can do in industry. I think people oftentimes narrow things down. So when you think about your values and then you think about things you're good at, what you like to do, keep this in mind. Yes, I see a hand raised. That's fun.
Yeah, thank you. This is terrific. So one question I had, you may have answered it because I had to leave, but I think one of maybe the myths you can disavow people of is this idea if you go to industry, you won't have autonomy over what you will work on. Hey, could you comment on that?
Yeah, it's funny. I usually respond in a way that maybe surprises people. You have limitations in academia as well, right? Like, you have grants that you need to show progress on that grant. You just can't wake up one day and go, I'm going to just start doing something really random. You'll run out of money pretty quick. So I think there's always some guardrails. Different companies have different levels of flexibility. At the end of the day, companies have to make money and they have goals that they need to hit. Different research groups have a lot of flexibility. My group has a ton of flexibility. So I think it really is role dependent and it varies a lot. I will say if you're talented and motivated, there's oftentimes a lot of flexibility for those individuals that have a plan and can demonstrate how it would be useful for the company. You just have to ask. A lot of people don't ask or they wait for permission and you kind of just have to demonstrate it yourself and go out there. Like don't drop $1 million on a project, but I think there's more flexibility. So I don't think it's as limiting as academia is in a lot of respects, if you really think about it.
Right. So the other question I had, and I think in your case, you described a situation, and I know Joe Gleeson very well, so I can understand also how the environment was optimal to find your passion. what do you suggest if people are doing their postdoc and they're having trouble finding their passion? What's your recommendation there?
Yeah, I hope I don't get in trouble for saying this. So I think life is short, and if you're miserable, you should do something else. I think if you have hopefully a good postdoc mentor, that can be done within the lab and you can explore different things and there's flexibility. Some labs don't have that flexibility, right? Like you have your own, everyone has their own deadlines and grants to apply to. I think it's worth considering trying other things. I think too often, as I mentioned kind of before, this is especially in academia, we get kind of focused on a path and really don't see a way out of it. And I've heard people talk about being miserable with their job and like, well, just do something else. And that is oversimplifying to some extent, but in some extent it kind of isn't, right? So I think it's important to really kind of think out of the box, do your own reading, look at your own hobbies, like don't make your entire identity that one position in the lab working on one project. Take a step back and think as a scientist and just as a human being, what are other things that I like? And I think that's an actual act, like an exercise people really need to do to think about it. But I think people are too often scared to make changes. I know if you're in the country on a visa, and so I know there are other considerations there that I'm very respectful and mindful of, that they don't have the same flexibility in employment. And my hope there is that you just have a strong community that's open to allowing some exploration in other places.
Great, that's terrific. Thank you so much.
You're welcome. Okay. So if you have interest in an industry job, the next couple of slides will just be kind of my recommendations to how to get a job these days. I have done a lot of hiring over the years. The first thing is don't go in with like a 10-page CV, 2 pages max. You need to convert your CV into a resume focused on what you've actually accomplished. And I think I touched on this a little bit before, highlight transferable skills, poster presentations, lectures, excellent communication skills, supervising a grad student, management skills, project lead, you put together preliminary budget, you need to mention that. Like, that's great. You're emphasizing your project management skills. Did you get a grant? You're showing that you're influencing others, and that's probably the ability to influence others, especially people who don't work for you and don't report for you, is an incredibly important skill in industry. Something I had to learn really quick, that for me to be successful, I need a lot of other people that don't directly report into me to buy in for what I'm trying to pitch here and to work together. At a place like at Illumina, it's not a hard sell because we're a very mission kind of goal-oriented group. But there are other places where, or other times it can be tricky. So influencing others is really, really critical. Method development and problem solving is just as important, if not more than important than publications. I've never made me personally have never made a hiring decision based on someone's publications for what I do. But if you demonstrate that you have, in the case of a bioinformaticist, you've put stuff in GitHub, you've developed tools, or you can speak to how you've developed tools as a postdoc, those things are super, super helpful, and just genuine or general problem solving. And the other thing I would do is, ask other professionals to review your resume. It helps a lot. If you're someone that's looking through these right away, I can spot immediately, oh, that's probably something I would throw out because it's missing XYZ, or something that I would look a bit further. These things all help a ton. I would, keep your LinkedIn account up to date. It's really the most common thing that recruiters and people are looking at. There's jobs on LinkedIn. Reach out to your friends and colleagues in industry. A lot of positions are word of mouth. They're never put out, or they're put out with a weird job description, and then you talk to them and you realize it's actually something else. So I think being open, even if you're not looking for a job now, if someone says, hey, can I chat with you? I'm kind of interested, chat with them. Be open. The best time to find a job is when you're not looking for a job. There's no pressure involved, right? So I would just kind of keep that in mind. Just keep your options open. And that's academia or industry. If there's academic positions coming up, talk to those people. Don't just network, build relationships. You can see through kind of superficial connections pretty quickly. You don't have to match every single qualification to apply to a job, but don't lie about it either, because you'll get busted really, really quickly, and that's embarrassing for you. And I think it's also just if you're interested in a particular space like biotechnology, it's good to just kind of generally keep up with the trends in that space. It will help a lot when you actually do an interview. There's different websites that you can look at. Do your research. I think I go right into this. When you do an interview, make sure you know that company and the business. Because it shows you're really keen. Otherwise, it just lets you apply to a bunch of different jobs and try to find something that hit. Honestly, the most important thing to me is enthusiasm. If you're not enthusiastic, that I don't care how good you are. I'm just not interested. We really want people that are excited and motivated. You can always ask questions from the hiring manager, especially if they ask you to give a presentation. Sometimes a job talk, an academic talk, is not what you want to do in an industry interview. It is super helpful, and I did this when I applied as well. Ask the hiring manager if they want you to give a presentation. Go, what are you looking for? What would you like? Ask specifics. Ask a bunch of questions. In fact, if you don't ask questions in an interview, once again, it can come off that you're not interested. Ask those questions about work-life balance, culture. Ask those same questions to everyone you interview. Make sure they all line up and you're not getting drastically different answers. I think the key thing is to be confident. So that's my spiel. I feel like I've taught for a long time. Happy to take any questions that people have, or if you're tired of listening to me talk, that's also okay.
All right,
I think
Alex,
that's
that was
great.
great. Thank you so much. Please, if you have specific questions for Dr. Crawford, we can pop them up in the chat or just unmute yourself and ask away.
Can I ask a quick question?
Yes.
So one thing I've been asked, and I don't honestly know the answer, is, so if you have a postdoc who thinks maybe industry is the way they want to go, and then they find an industry job and they do that job and they decide that's maybe not where they want to be. What is, in your experience, what is the ability and potential to move back then into academia if they decide that that's the way they wanted to go?
Yeah, that's probably a better question for those in academia than for me. I have known a few people that have done that. They usually make those decisions pretty quick. They're usually not in industry for like 10 years and then decide to go back. I think it's kind of the same way to get your industry job. It's going back and networking, highlighting. So you may not have the publications, but you need to highlight how you would add value to that institution. There's people I know in industry that start with adjunct professors and they teach, and that's how they kind of have an inroad into a local university. And there are actually quite a few that do that. I think if that's what you're leaning towards, I think it would be like migrating to somehow become ingratiated in that community again. Participating in conferences. It's networking again.
Right.
But I don't know, Edmund, if you had better advice for that.
I mean, I don't know that many examples of people going back and forth. There are a few, but yeah, I don't necessarily have the experience with that or the advice to speak on it. I think the thing that caught me that you said also, that program officer from NIH and one of our previous things said, was to basically take advantage of these opportunities and network and really listen and engage with people that aren't necessarily in your field, the people that are in industry, and just learn more about it, and don't be afraid to take that risk of stepping outside your comfort zone. If you listen to them and you say, hey, this is actually kind of a really cool path that you could take.
I also think I like to amplify something you said, and that is, I think there is a lack of clarity on the part of many postdoctoral fellows of when they're ready. And I think the thing that you said about actually starting to look maybe well before, or being open to looking before you actually feel like you have the requisite set of skills, is actually a really good idea, because there is a stochastic, as you pointed out, there's some sort of stochastic probability that you're going to align your interests with a job. And that may not couple with when you think you're ready. So starting to look well before you think you actually are ready might actually, I thought it was a really good piece of advice. And being open to actually a job that might allow you to pursue your passions. So thanks again for that.
Thank you.
All right, there's a question from the techs and postdocs.
Hi, my name is Ayushma. I'm a technician here. My quick question is, when you mentioned when you started a new position and you had that internship period, right, when you were kind of not placed into a box or a position and you were exploring many roles, how much do you think that was because of your personal motivation to like explore? And also, and how much do you think that was also the environment and the encouragement to do so? Is that a common thing you see in industry, or was that specific to your company?
I don't think it's super common. I will say Illumina is very interesting place in the sense that they encourage and they're open to the exploration within the company. So if I had to do the same role that I was hired originally, I probably would have left Illumina a while ago because it really wasn't what I enjoyed doing. But I've been there 10 years. And so Illumina and probably a large company, I think that's something be even a larger company side, although I'm not sure, we have an internal hiring portal and people switch jobs. It's not that uncommon of a thing. And in fact, I was relaying a story earlier that someone that has started in our group as a scientist decided he wanted to go into finance, which I was like, dude, why? But whatever, he liked it. And now he's moved on and he's like a SVP of another company and it's like climbed in. Totally, who would have thought? But it's something he just wanted to be open to and try. So there was a position within the company. I think it helps if it's a larger company, you just tend to have more positions. And then you apply just like anyone else would go through that process and then you can get hired. So that internal moving, I think probably saves the company money in terms of recruitment. Like they want to retain you. Like you don't want to, what was it called? I think it's called like regrettable turnover or something like that, where you have good people leave the company. Even if they do something different, if they're really talented, you want them to stay, you want to retain that talent. So this is something, a benefit that Illumina has that I think is absolutely fantastic. It's a great aspect of the culture. I can't speak to how many other companies that offer it, but I would imagine there are those opportunities available. I mean, I'll be the first to admit, I got really lucky. I had really, really great mentors. In fact, if I was to give another piece of advice is find really solid mentors that support you. I still talk to those people today. We're really good friends. They still look out for me. It made all the difference in my career trajectory. All the difference. I was given opportunities that I wouldn't even known had been available if it wasn't for those kind of interactions. And then pay it forward. If someone does it for you, you better make sure that you look out for other people and do it for them as well. And I do try to. So it's out there. You just got to ask. You never know until you ask and push for it. And we love technicians. I love people that like to stay in the lab and do bench work. That is not always the easiest thing to get. And if you love pipetting, we got jobs for you.
Don't just have robots for that, huh? Can't just control them with AI now, and
could never replace the human hands.
there's a few you could wish
For now.
For now.
That's
I have
really
a
cool. I mean, I
question.
think I know.
I have a quick.
Oh yeah.
Oh, sorry to interrupt. First, thank you for the talk. And I wanted to ask, like, from your experience working in the industry and seeing people switch from the academia to the industry, probably, do you think that there is like individual variations? People can do the switch right away. People take their time, or do you like advise? Like, is there a general advice for all, or do you think that there could be an individual variation for the timing or for the direction that they choose to pursue?
It's very, I think, individual specific. I've definitely seen people come in from academia that struggle to transition to the culture and industry a bit, kind of the pace. And one aspect, honestly, is the collaborative nature of it. Sometimes people want to do everything. I'm like, no. Like the bioinformaticist down the hall that got his PhD in this, he's going to do that. You do the thing that, that you were kind of hired for. Like there is some adjustments that I think that's probably some people pick up quicker than others, especially about things that I think the biggest one is time is money. That's not necessarily true in academia. Money is money, and you're more limited in resourcing. But to some extent in industry, you look around and it's like, how many people's salaries are we paying for this meeting? Is it worth it? It's kind of a little bit of a different mindset. There's people that come straight out of undergrad that go into industry that do well. There's others that come from postdoc. There's others that come from grad school. It really, really, really depends on the individual and then the group in which they're applying to. I don't think there's a universal rule.
Thank you.
So Ali, for you, right, you said you kind of stumbled across Illumina at the career fair at UCSD. I mean, but you were in your postdoc, you were there for 2 years. I mean, did you feel yourself that you were ready for that sort of jump, or was this just...
No, not at all. I mean, it was kind of, it came up and I was like, well, I guess I'll interview to see what it's about, right? They have a really nice little like coffee bar, and I was like, I want to get a good coffee and get a treat and whatever. Really nice, facilities. I was curious. I wanted to see it. And then when I got the offer and I met the people, that's the biggest thing. Like I really gravitated towards the people that I interviewed with. They were fantastic. Tim McDaniel was at Illumina at a time. He has, I think he was on a few podcasts. His mother, unfortunately, passed away from cancer, but I think she was probably one of the first participants where they used sequencing to try to come up with therapeutics for her. And he took time off at Illumina and worked at a company or an institute called TGen to try to come up with solutions. This man is so passionate about what he does. We used to get annoyed at group meetings, like, you can't make us cry every week. It's super awkward, but very emotional, so invested. I mean, after that interview, any question I had, I was like, oh, I gotta make this move. I think this is kind of like maybe potentially a once in a lifetime type of, opportunity to contribute to this space. So no, I didn't feel ready. I just kind of knew when I saw the opportunity, I was like, I think this is worth the leap. I was actually really happy in Joe's lab. I was happy as a postdoc. I did not leave disgruntled by any means. I still interact with Joe actually quite a bit in different areas, which is kind of fun. And I think his group does great work.
Thanks. More questions from the group? All right. I think that the I Know project you talked about, it seems really, you touched on a little bit that AI tools are going to be critical for that space, and trying to dive through the literature, trying to dive through sequencing and figure out really what is linked to these specific diseases. I think that's probably the next, 5-10 years of where that project goes. It's got to be in developing those AI tools. So I'd imagine Illumina probably, you said that you've got some already through Illumina, but do you partner with other institutions, or do you kind of build this in-house?
A lot of that has been built in-house. We have an AI team, super talented group of individuals that have been looking at it. But the cool thing about it, at least for now, and I hope it stays this way, is those tools are really, they're available to the public. I think there's a license for commercial institutes, but in academic institutes, I'm pretty sure it's free to access. And so it's I think when AI and all of these sort of generative AI tools, garbage in equals garbage out. You want good data in to get good data out. So I think the more we encourage collaboration, and they also operate in large respects like an academic group. They read papers, they have journal clubs, they're looking for new techniques. I think they're very, very open for collaboration. It's a very, like I said, our groups are a little unique in the industry, but super rewarding and a lot of fun. But yeah, that's the future for sure.
Yeah, it sounds like a wonderful environment. I mean, you've been there for 10 years. I mean, if the internal transfers in order to retain people, it sounds like it probably has relatively low turnover for the industry, and it seems like it's a great environment.
It has been.
All right, I'm going to stop the recording now, somewhere here.